In every single cell of the body, there contains an entire copy of your genomic DNA. Every single cell except for the blood cells, because the blood cells, they just pick up oxygen and drop it off. However, in every single cell, from your skin cell to your lung cells, to your bone, to your nerves, all of these have an entire copy of your genomic DNA. The only difference is that there's a distinctive way these are organized. There's two big things to note. One is that there's 3 billion base pairs of your DNA encapsulated inside every single cell. If you were to stretch out that 3 billion base pairs of DNA linearly, it'd be about 2 meters long, or about 6.5 feet tall. I'm 5'2", so it'd be even taller than me. Yet somehow this is wrapped and organized into the 10 micrometer size small of your nuclei. The second big thing to note is the fact that the way that this is actually organized allows for the distinguishment between your hair cells, your lung cells, your nerve cells, and your bone cells. And that's a good thing, because the cell needs to be able to store away all the genes that it doesn't need in order to carry out the functions of a lung cell, for example, and leaving only the genes that it does need out. Think of the way that you storage your clothing or decor. You don't have winter coats out during hot summers, and you don't put out Christmas trees in May. For the same reason, your cells need to be able to storage this away and properly, so that way it can carry out the functions that it needs. Hi, my name is Karla Isabel Medina, and I'm a PhD candidate in the Interdisciplinary Biological Sciences program with the Department of Biomedical Engineering here at Northwestern, where we converge biology, optics, and physics-based modeling in order to understand how the structure of your DNA shapes and forms and organizes in order to distinguish those different cell types or functions. We understand that there are confounding differences between the structural organization of that genomic DNA, which allows for the different cell types that we observe. We want to now explore this in cancer cells and to specifically understand how this organization changes with disease. I apply this work to cancer, and here's a quick scenario to highlight how and why. Think of a healthy cell and a cancer cell. If you treat these two cells with the same cytotoxic stressor, such as a chemotherapeutic, it needs to be able to modulate that structural organization or those domains of chromatin or DNA in order to respond effectively and gain survival. Oftentimes, though, what ends up happening is that the healthy cell, these die off. And the cancer cells, they're able to modulate their domain organization of DNA in order to survive. AKA, they're able to express the genes that it needs to and access them when it needs to in order to survive. This added survival allows cancer to keep coming back. These cancer cells that live are called cancer stem cells. And we call them cancer stem cells, or CSCs, because they can actually give rise to new tumors and allow those tumors to spread via metastasis and go all throughout the body. Another feature is that they also express stem cell-like properties, which give them an added advantage as they can grow out those new tumors and metastasize and travel to distal regions, as I just mentioned. My doctoral aims to understand how the structural organization differs between the cancer stem cells. This altogether will help us understand how the genomic DNA and its organization is actually facilitating their survival. By uncovering this, we can also develop new ways to target these cancer stem cells and eliminate cancer for good. Our group, along with others, are working in an emerging new field called physical genomics, where we explore how the physical genomic organization of the DNA facilitates the observed outcomes that we see. Altogether, we converge molecular biology with optics and modeling approaches in order to elucidate the structure of chromatin, understand how its structural organization influences the cellular plasticity we see, as I mentioned in cancer stem cells, and further exploit these modalities in order to understand how the domain organization of our DNA relates to the disease outputs that we observe. To learn more about our research or our nanoscale imaging platform, please visit our website to see how we apply these techniques all together to help the patients in need.